Hi everybody, welcome to topic 3, where we're looking at, we're moving beyond water and the cell to water and the plant. And some similar concepts apply, but now we're looking at it from the nexus between the soil and the plant, and then the root zone and the canopy of the plant, and then the canopy of the plant and the atmosphere. So we're looking at what forces drive the movement of water from the soil, or within the soil I should say, into the root then to the cambial tissue, up to the canopy, into the leaves, and then from the leaves this water is converted back into a gas and lost to the atmosphere. Water balance of plants is an incredibly challenging thing for land plants to do, namely because water is generally limiting in most environments, especially those in the Mediterranean climate here in the southeast of Australia. And the problem for plants is that their foodstuff, carbon dioxide, which through photosynthesis they convert into carbohydrates, that's absorbed from the atmosphere. And to do that they have to open the pores in the leaves called stomata. Every time they open their pores, carbon dioxide is absorbed, but also water is lost. Likewise, the mechanism that plants use to shuttle nutrients in the soil, which is the basis of topic 5 and topic 4, so the movement of nutrients from the soil up to the canopy relies on water movement to be the transport agent. All right, and that process of movement requires, by definition, water being lost from the leaves to allow it to take place. So let's have a look. Let's look at the soil first up. Soil has a number of different properties, or we have a number of different types of soils. On one hand, we have the sandy soils, which have the largest particles and the largest spaces between each soil particle. Because they're such large particles, there's actually a low surface area per unit weight of soil. Whereas clay soils, on the other hand, which have a much finer particle, has a much larger surface area per unit weight of soil, and this is important, which we get to. There's small spaces formed between these smaller particles, or pores, as well as organic matter which accumulates within these clay soils can also lend its own properties. So after watering, when water hits the surface of the soil, it moves down between the spaces in the soil, either displacing or trapping air in pockets. And through the capillary action of water, the smaller pores are filled first and the larger pores filled later. We'll look here at how the properties of soil in particular changes the movement of water within the soil, how that makes it more difficult or easier for a plant to obtain water from the soil. And later on we'll be using this in later topics to see what this has to do with availability of nutrients as well. So first of all, we remember back that water potential, or Psi-W, is composed of 3 facets. There's gravitational potential, and in the soil, if we're talking about from one area of the soil to another, the gravitational potential doesn't change. But when we start moving up the plant, moving up vertically in the atmosphere, let's say, then the gravitational potential will change. But here, the soil water potential and differences in soil water potential, gravity doesn't play a part. It's subtracted from the equation because it's the same. It's all at the same level. So we've got gravitational potential, which partly makes up water potential, and then there's also the osmotic potential, the potential which is provided by the dissolved solutes in the water within the soil. And generally that's very low in soils because there's a low concentration of dissolved solutes per unit water in the soils. Apart from extremely saline soils, which can get the osmotic potential can get quite negative, but generally in non-saline soils, the solute concentration is around negative 0.02 megapascals, right, the osmotic potential. The other component is hydrostatic pressure. So water potential is the potential due to gravity, the potential due to pressure, and the potential due to solutes. The hydrostatic pressure within the soil, in extremely wet soils which are saturated, the hydrostatic pressure is 0. But as soils dry out, the hydrostatic potential actually becomes negative. So let's look at why that is the case. Well, water, due to the hydrogen bonding that it does between itself, has high surface tension, which is attempting to minimize the interface between air and water. So that's why it forms droplets. So it naturally wants to minimize the contact with air, but what also happens is, due to the adhesion properties of water, it binds to soil as well. So as the soil water decreases, water recedes from the air-water surfaces by curving around the soil particles. And as water becomes curved around these soil particles, it develops a negative pressure. So the stronger it adheres and the more curved its surface, the more negative hydrostatic pressure is what builds up, or tension, in other words. And this is what drives movement of water through the soil by a mechanism which we call bulk flow, which is the mass movement of molecules, in this case in response to a pressure gradient. Right, so bulk flow, when we're talking about bulk flow in soil, that's water moving from an area of low or high hydrostatic pressure, where there's lots of water, to areas of low hydrostatic pressure, where there's less water and it's adhering to the soil particles and curving around them. So what happens is, a plant sitting in the soil is drawing water out from the soil, and as it does that, there's less and less water there, and what water does remain begins curving around the soil particles. And that creates a pressure gradient within the soil, and water will be drawn towards the root zone because it's actually being drawn towards these areas of negative hydrostatic pressure. The rate for which this water moves through the soil is known as the conductance, the hydrological conductance. And conductance is a concept which we'll be coming back to repeatedly. And conductance is just essentially the rate for which water or a gas flows through something. So we might talk about stomatal conductance, and that's the rate for which gases flow through the stomata. Root conductance is the rate for which water moves through the root zone. Xylem conductance is the rate for which liquids can move through the xylem. And conductance is always down to two factors: the size of the pressure gradient, right? So the difference between the area of high hydrostatic pressure and low hydrostatic pressure, the bigger that difference, the quicker water will move. And also the ease for which the water can move. In other words, the size of the conduits or tunnels or pores for which the water can move through. And if you think about it like a dam, it makes sense. You have a dam, heaps of water is being stored behind that dam, building up all this positive energy, this positive hydrostatic pressure which is being forced upon the dam wall. The river below essentially has negative hydrostatic pressure, but it's a gravitational force. If we have a small tube or pipe coming from that dam and a large diameter pipe, more water will flow quicker through the large pipe compared to the low pipe. Likewise, a pipe at the bottom of the dam wall will flow quicker than one at the top of the dam wall because there's more pressure. Water is physically absorbed by the roots once it gets to the area around the root zone by the root hairs. Nutrients can be absorbed by different parts of the root, and different nutrients get absorbed by different parts of the root zone. But water itself is generally absorbed through the root hairs. And this can be a problem when we have shallow rooted vines in our vineyard, and we compact them, maybe due to mechanical harvesting. And our root hairs, we can lose up to 95 to 98% of our root hairs, and all of a sudden, one of the main mechanisms for water absorption is gone from our vines. Likewise, if we have a perennial crop or an annual crop even, the same thing applies. If we compact our crops through maybe livestock, then we're reducing their capacity to absorb water. Water gets into the root through a number of ways. First of all, it can go through the symplastic and transmembrane pathways thanks to the root hair, or it can enter the root zone, again still via the root hair, but through the apoplastic pathway. When it goes through the apoplastic pathway, it's just passing along through the cell walls of the root cells. So first of all, sort of enters the cell wall of the root hair and then moves from one cell wall to the other towards the stele in the center of the root. All right, so in the apoplastic pathway, the water never passes through a membrane. The transmembrane pathway, it enters through the membrane of the root hair, and then through the membrane again into the next cell, and then through the membrane again to another cell, physically through the membrane, all the way into the center of the root. Through the symplastic pathway, it enters into the root hair, and it has to go through one membrane to do that. But then it passes through those intercell channels called the plasmodesmata. And remember, they're the channels that link between membranes of neighboring cells. Right, so the symplastic pathway, we don't often talk about the transmembrane pathway, but we often talk about the symplastic and apoplastic pathways. Symplastic, I remember sim as in, it means it goes into the membrane and passes through from one cytoplasm to the next, and apoplastic is outside of the membrane through the cell walls or the extracellular space. I have no clever way of remembering apoplastic being outside. I just remember it's not sim, which is in. As the water's moving through the root, and I shouldn't have clicked past that slide as quickly as I did, but water in the apoplastic pathway gets stopped by this hardened band of tissue called the Casparian strip. And that prevents any liquid in the apoplastic zone from getting into the xylem. That means the plant only allows liquids which have made it through a membrane, at least one, to get into the conductive tissue of the root. All right, so that's just one mechanism it uses to try and moderate what actually gets into its root zone. For example, if it didn't have that, all the salty water that enters the root zone through the apoplastic pathway would just make its way straight to the xylem. However, by having the Casparian strip, that salt actually has to get into a membrane at some point, and once it does that, the root is very tricky about it and starts sequestering it in its vacuoles. And so that's one mechanism that plants have to tolerate salt. So once the water's in there, it's passed into the root zone following a gradient of water potential. So it's made it to the root zone following a gradient of hydrostatic pressure. But remember, the soil generally has low osmotic potential because it's got low dissolved solutes. Well, the root zone, on the other hand, the cells are loaded with different solutes, like carbohydrates, for example. That reduces the water potential of the root zone below that of the soil water, and that's why water will move into the root zone. If transpiration is low, so no water is being lost from the canopy and all this water is entering the root zone, actually builds up a lot of positive hydrostatic pressure. We're stuffing in a lot of water into a small space. This can be important for perennial crops that go through dormancy, like apple trees or grapevines, because as winter's breaking, a lot of the starch stored in the root zone get reallocated or get converted back into sugars. That drops the water potential of the root zone, leading to a flux of water from the soil into the root zone. However, the water's got nowhere to go because the plant doesn't have any leaves yet. So this water just pouring into the root zone, building up the pressure, but it's got nowhere to go. So if you cut one of the branches, all of a sudden you've relieved a lot of this pressure. So all this water is being forced into the xylem, but it's trapped in the xylem. It's got nowhere to go. If you chop one of the branches, all of a sudden there's an opening, and it relieves that pressure, and that's why you can get this exudate coming from wounds or pruning, if you do late pruning of dormant trees. The pressure can build up so much, even in plants with leaves, if transpiration is low, that you can actually see it coming out from the edges of leaves. And on this slide, I believe that's a strawberry bush where that's occurring. So the xylem, is a recap from topic 1, composed of 2 tracheary elements. They're called the tracheids, which are the primitive form of xylem tissue. They don't allow linear or straight up water. The water has to take a circuitous route up the tree, and it goes from one tracheid cell to another via these lateral pit cells. Vessels, on the other hand, are more highly evolved xylem cells, and they allow vertical movement of water through these perforation plates. These perforation plates play an important sieving role, especially preventing the movement of air bubbles, but they still allow much quicker movement of water. And you think about that principle of conductance, remember, the rate for which water can move is at least partially related to the size of the vessel. And so here we see these vessel elements making it much easier for water movement. How does water go up the xylem? So, all right, we've got this water moving in along a water potential gradient from the soil into the root zone, and then it has to go into the xylem, but the xylem is full of water and very few solutes compared to water. So how does the water get in there? Well, the only way that the xylem can lower its water potential below that of the root cells is by reducing its hydrostatic pressure. And it does that in 2 ways. First of all, it's because water's getting sucked out from above. All right, so water's getting sucked from the leaves, which is drawing water from the xylem, creating a negative hydrostatic pressure, a pull, a tension. Add to that the ability or capillary action of water, such that one water molecule binds to another, and that allows it to build up really high tension in these xylem vessels. So, as leaves are removing water from the system, there's a pull on the water in the xylem tissue, and that pull makes the hydrostatic pressure drop in the xylem cells, makes it more negative, and that makes the water potential more negative, and that's what allows there to be a gradient from the root zone into the xylem. So our water's got to the top of the xylem, and then somehow it's got to get into the leaf, and so again there has to be a reduced water potential in the leaf compared to the xylem. Well, the difference in water potential is very low in the leaf. Remember, it's got positive hydrostatic pressure within the cells, but outside the cells, it's got to get to the cell walls to then enter the cells in the leaf. And they achieve this to get water from the xylem in towards the mesophyll cells of the leaf by the orientation of the microfibrils, the cellulose microfibrils in the cell wall. They form these triangles which actually suck water in. Remember, water in the pores of soils makes that curvature to try and reduce the air-water interface. Well, the same thing happens with these cellulose microfibrils. It's quite ingenious. So the microfibril angle actually draws water from the xylem, and that produces a negative hydrostatic pressure, which draws the water in. And you can see that the radius or the angle of the curvature of the space actually increases the hydrostatic pressure. So if you drop the radius 50-fold, you increase or make the hydrostatic pressure 50-fold more negative. As I said before, sometimes we get cavitation or embolism in the xylem where gas bubbles form. This can occur when xylem tissue freezes, which forces gas out of solution. At other times it can be when there's so much tension on the xylem column that it actually snaps. So the tensile strength of the water is below that of the tension being, or the pull being forced onto that xylem column, the water column, and the water column actually breaks, and we get these gas bubbles. And the beauty of the pit membranes and the sieves on those xylem cells is it prevents gas moving too far. Because if we get these gas bubbles, all of a sudden that xylem can no longer drag water up from the root zone up to the leaves. And so these cavitation can be extremely damaging. So as this water is continuing its journey from the root zone up to the leaf, some of the water is then absorbed by the cells, the mesophyll cells in the leaf, to drive photosynthesis and maintain turgor pressure of the leaf. But of course, other has to be lost to the atmosphere. A lot more of it has to be lost to the atmosphere to maintain this transpiration, to maintain this movement. So what happens is the water gets dragged towards the cell walls by the negative hydrostatic pressure of those cellulose microfibrils, or the adhesion to those. And once it's there, it then evaporates into the air spaces within these spongy mesophyll cells in the cell there. And once it's in there, we have a highly saturated environment. So we've got an extremely humid, or gas which is highly saturated with water. The outside of the leaf has a really low water vapor concentration because it's the atmosphere, so there's heaps of air and not a lot of water, even on a humid day. And so instead of having a water potential difference, we actually have a water vapor gradient. So we've got high water vapor concentration inside the leaf and low water vapor concentration outside the leaf. And that's what drives water movement through the stomata, this final movement. This is a really short path length, and water moves through it very, very quickly. However, the resistance to the movement is very large because, first of all, there's this waxy cuticle which prevents a lot of water movement through. And then these stomata here, which allows water to go through very quickly, but they can open and close. So again, the rate for which the water will leave the leaf depends on the difference between the water vapor concentration inside the leaf and that of the air. So the only thing stopping this water moving along this concentration gradient is, one, the concentration of that water vapor, of course. But two, remember, it's the other one. So one is the gradient, the other is the size of the openings. And as I said, the way, apart from getting through the cuticle, which is extremely difficult for water, the only way this water vapor can leave is through the stomata. So one way it prevents it is by opening and closing the stomata. This is called leaf stomatal resistance. By closing the stomata, you're increasing the leaf stomatal resistance. And the other way is by making the air around the leaf humid. And that's called the boundary layer, that area around the leaf. And so by increasing the humidity of the boundary layer, you're increasing the leaf boundary layer resistance. One way plants do that is by reducing airflow. It's pretty much the only way they can do it. Some plants do this by hiding their leaves. Others do it by only growing in sheltered areas. Other ones put hairs on their leaves to reduce airflow across the stomata, physically having hairs there. So most water moves through these stomata, and here's a fantastic micrograph of stomata. What we have is guard cells, these two cells bounding the hole in the middle, which is where the gas is exchanged. We have these subsidiary cells on the outside, which help the mechanism of these guard cells, which we'll go through. And then next to that is just the epidermal tissue of the plant. So these openings between the guard cells is where gas is exchanged. And so when a plant wants to reduce the amount of water loss, it closes its stomata. There's main two types of stomata. There's dumbbell-shaped ones, which are found in grasses, and kidney-shaped ones, which are found in dicots and all the non-grass monocots. And these guard cells have extremely thickened cell walls, which allows them to maintain a high hydrostatic pressure. And what essentially happens is ions are pumped into the guard cells, reducing their water potential and leading to water flowing into the guard cells. And when that happens, they expand, and due to a reconfiguration of their geometry as they expand, the stomata opens allowing for gas exchange. So this slide just describes more what I said, and here we go. The role of the subsidiary cells play that role of sort of being the bank for these ions which get pumped into the guard cells, dropping their water potential. So an overview, the soil plant atmosphere continuum, the transpiration, the movement of water from the soil up to the atmosphere follows a chain of ever reducing water potentials all the way up. And different elements use different parts. The root zone uses a low solute potential. The xylem uses a tension, so a low hydrostatic pressure. And then when we get to the leaf, some water is used for photosynthesis and maintain turgor pressure, and others is lost directly to the atmosphere to allow for that constant movement of water. And there, the rate for which the water is lost depends on the difference in water vapor concentration on the inside and the outside.